In this lecture, I'm going to talk about the complement system just in general and in brief. And as you know, the complement consists of more than 30 soluble and membrane-bound proteins. And the soluble, they are actually circulating in inactive form. And the complement system is a major effector of the humoral branch of the immune system. And as can be seen here, B cell, they express complement receptor number 2, which interact with complement 3D, and this will provide B cell with an up to anti-apoptotic signal. As can be seen here, this story actually it takes place in the germinal center, and this is antigen coated with C3D, and it interact with CR2 expression on the surface of the B cell and CR2 expression on the surface of the dendritic cell. So the B cell here receive an anti-apoptotic signal from the dendritic cells. So now let us discuss in brief functions of a complement system. So the complement system has a capacity to lyse microorganisms by means of membrane attack complex. As can be seen here, the membrane attack complex has a capacity to remove phospholipids from the wall of infected cells or from the wall of the viruses, and this actually will lead to disruption of the osmolarity, and the fluids they enter and cells they get swollen and eventually they rupture. And also one of the functions of the complement system is that it promotes phagocytosis by means of opsonization. As can be seen here, this is a bacteria coated by complement component, and this when they bind to their specific receptor on the surface of the phagocytic cell, this will help in their phagocytosis, are known as complement mediated phagocytosis. And also one of the functions of the complement system, as can be seen here, these are the neutrophils. They express receptors CR1 and CR3. So when microorganisms binds, coated microorganism coated with complement component, when they bind here, this will lead to degranulation and release of mediators that may kill the microorganisms. And as can be seen here also, it is very important clearance of immune complexes from the circulation by means of red blood cells, as they express complement receptor number 1, and these red blood cells, when they bind the immune complexes, they bury them in the liver and spleen to avoid autoimmune disorders like, or for example, systemic lupus erythematosus or glomerulonephritis or vasculitis. So now I am going to talk about the different or the three pathways of the complement system: alternative, classical, and lectin pathway. Actually, this is alternative pathway, so it does not require antibodies, and it requires only, for example, non-self or antigens on the surface, non-self antigens on the surface of self cell. So this is self cell express non-self antigen. So this will be recognized by spontaneously cleaved C3. C3 will actually spontaneously cleave into C3A and C3B, but at very low rate. So C3B it binds to non-self antigen on the surface of self, and also it binds to polysaccharide on surface of bacteria, and it binds to snake venom. So the binding of C3b actually will activate factor D and factor B. So this activation will lead to cleavage of factor B into BA, BB. So BB will bind to this C3b, and this will lead to formation of C3 convertase, which act on the circulatory C3 and cleave it into C3a and C3b. So C3b will bind, and this will lead to formation of C5 convertase, which act on the C5, and C5 get cleaved into C5a, C5b will bind. So no need now to cleave more complement components as the machinery now is on, and this will lead to formation of membrane attack complex, which is very important for killing of microorganisms by removing phospholipids, and as shown above. So this is classical pathway. The classical pathway it differs from the alternative pathway as it requires antibodies. In case of IgG, we need two molecules of antibodies, but in case of IgM, one molecule can activate this pathway. So now this is antigen or bacteria or whatever non-self. It is captured by these antibodies, and then the antibodies will bind to complement component number 1 to C1q, which associated with these proteases C1r, C1s, and this will lead to activation of C4 and C2, and they get cleaved. So C4b will bind to C2b. Then this is known as C3 convertase, and this will act on C3, cleaved it into C3a, C3b. C3b it binds to this molecule, and this will lead to formation of C5 convertase, and this act on C5, and C5 get cleaved into C5a and C5b, which it binds to the remaining of the cleaved molecules. So no need now to cleave more complement components as the machinery now is on, and this is known as from C5 to C9. It is known as membrane attack complex, which remove also phospholipids. However, this is associated with formation of an aflatoxin C3a, C4a, C3a, C5a. C3a and C4a they stimulate release of histamine, and histamine in turn increase capillary permeability, vasodilatation. C5a is an chemoattractant, attract cell. So now we have attraction of cells, we have increased capillary permeability, we have vasodilatation, and this equal to the three major events that take place during inflammation. So activation of this classical pathway will lead to inflammation and formation of the membrane attack complex. Now I'm going to talk about the mannose binding lectins. These are actually proteins produced by the livers in response to some infections, for example, Salmonella, Neisseria, Listeria, Candida. However, this mannose binding lectin is very similar to C1q, but it is associated with serine protease 1 and serine protease 2, and here also it is associated with two proteases C1r and C15. So this association will act on C4 and C2 similar to this the effect of this molecule on C2, C4 and C2, and then C3 will get cleaved, C5 will get cleaved, then this will bind without cleaving, and this will lead to formation of membrane attack complex and some of the anaphylaxis. And this membrane attack, as can be seen here, it has the capacity to remove phospholipid and to create holes or pores on the cell, and this will disrupt the osmolarity, and it will lead to rupture of the infected cell. Thank you very much, Professor Ahmed Bola.